Cool. All right. And could y'all tell me if you can hear me in the chat, please?
I can hear you.
Awesome. Thanks, Zhang. Cool. And thank you everyone who responded in the chat. All right. So howdy everyone. Welcome to the first Dr. Garcia Microbiology SI session. Super excited to be here today. Welcome in. Super excited to be here. And yeah, so if you've been here for a sec, you may have heard me talk about the itinerary, but basically we're going to be, what was the first thing I was going to do? Yeah, we're going to go over the problem set. We're going to go over the problem set, and then we're going to talk about, no, we're going to go over study habits. You didn't listen. You didn't listen. You weren't listening earlier. It's fine. No, so we're going to go over study habits or like study techniques and stuff like that. Then we're going to go over the problem set. So I've done it already, but I'm not allowed to just give you all the answers. So I'm going to have you all work together. And then after that, I'll just be like listening in on what y'all want to get out of the SI sessions. I haven't really planned anything in terms of like the rest of the sessions throughout the year, because I wanted to hear what y'all wanted to do. So we'll get into that later. We don't have to worry about that right now. But so yeah, you guys have been in class for about a week or so. So you may have noticed this is a very like PowerPoint heavy class, right? And the nice thing about Dr. Garcia is that when he says everything's on the slides, like he means it. Like you won't see a lot of test material hidden inside the figures. Like I know in some of like my other classes that I've taken in the past, they'll say like everything you need is on the slides, but then it'll be like the blurriest piece of crap in like a figure that you needed to know, and then it doesn't really count. Oh. I think your mic's on. Sorry, all good. So yeah, it just wouldn't be helpful. But Dr. Garcia is super nice about it. Like he'll highlight important things and then he'll put it in like actual text instead of like hidden in the figures. So that is, I guess if you can call it a study material, that will be like the most important thing. If you want to study, if you want to cram, that's the thing to go to. And then immediately after that, in terms of like importance, you'll have your problem sets. So these problem sets, he says that, oh, they're just for you to know what you know what you don't know. He actually goes a little bit further. I think I remember from last year when I took him that he would actually add some of the problem set questions onto the actual exam. So obviously it helps to have done them, right? So that's always nice. Make sure you do the problem sets if you want to do well in this class. And I think because I know how important they are, my only plan for this semester, or my only two plans, one of which is to go over the problem sets each session. I'll make sure to cover all of them. I'll make sure to clear up any confusion and have y'all work together on them just to make sure everybody like pulls together like the most, like the biggest possible pool of knowledge, whatever. And then I'll be like correcting y'all if I think I have a better answer, stuff like that. And then the other thing that I know I want to do is, because I have his previous exams, I'm not going to obviously give you the exam questions because he might be reusing them. But I will make my own questions based off of like how he formats his. So it's like I'll be like my own little UWorld or whatever, if you know what that is. I'll make sure to make questions that are pretty similar to his that cover the important topics. So yeah, come to the sessions if you want those, because I will be going over that. What else? So yeah, you guys are probably like, since it's the 300 level class and you need 213 to take this class, you probably already have your own study habits, which is really good, because I'm not comfortable like sitting here telling y'all that you need to learn using flashcards or like cramming the night before, or like whatever. But what I found helpful, because it is like really big on memorization and stuff like that. Shoot. Sorry, let me respond to something real quick. What I found helped me the most is Anki. So it's nice like having like the big concepts refreshed because they also do build on each other a little bit. What else? No, I make my own. So you could do Quizlet as well. So Anki and Quizlet. So Anki it's spelled A-N-K-I. Hey, are you here for microbiome? Yeah, so there was a huge ass class in there. Yeah, I'm sorry. I should have texted in the group me that we would be in here. And honestly, I might end up just releasing the recording of this SI session because I feel bad with the rain and like the room change and such. But anyways, in terms of study tools, Anki and Quizlet, just like flashcard systems, whatever works for you, and like spaced repetition, that helps really a substantial amount. If you're good at cramming, it helps in this course a lot, more so than like biochem or ochem. So that will help you. And just, yeah, like the lecture slides and the study questions will always be good for you. Yeah, so that's as far as study tips go. Does anyone have questions on like how the class is structured or like other things like that? Shoot, someone typed it in the chat as well. Should have been looking at that the whole time. Another thing I want to bring up is that, sorry, another thing I want to bring up is that lab will get you. Like I managed to, and I don't want to sound like pretentious, but like I did well on the lecture exams, but the lab midterms will fuck you up. Like I think on the first one I got like a 62 out of 100 because it's just like it's super application based, and then you need to know all these like very small differences between like different tests and stuff. So and maybe that's because I haven't really put in as much effort into lab practicals and lab exams like in other classes and stuff. But I think the lab exams are harder than the lecture exams. So just, I guess the only tip I can give you, since I didn't really do well, so I'm not going to tell you what I did, but just study in advance for those, take them seriously because they will take you down a letter grade if you don't do well. No. Well,
my lab TA like mentioned that?
I think they may have. The questions of this are similar in the sense that, for example, like a question will be, this is a picture of a petri dish, and then this is what it's been treated with, and stuff like that. What is it or what is it not? And like you have to go through like a flow chart in your mind of like whatever the heck's going on, and then you need to find out what kind of bacteria or whatever it is, which was really challenging for me, and it might not be for you. All I'm saying is that play it safe. Take the first lab practical seriously. Well, it's not a practical. Take the first lab exam seriously, and then if you find it's easy, then you can pull back on the throttle a little bit. But yeah, that's honestly all the advice I have off the top of my head for this course. But then yeah.
Is it like
So sorry, they say that again.
are the exams more like giving a picture and like identifying them, or like question based like right now?
Yeah, so the question was, if y'all can hear it, because I'm not sure yet. I haven't looked at the recording, but the question is, are the questions more like identify this organism, or just like a straight up question like what does this do or what does that do? I'm assuming that's what you're asking. It's more of the first one. And they will give you like freebies, but most of them are application based. Like they're pretty heavily application, which is why it's more challenging. But luckily the lecture exams are more like, what is this and what is this not? And it'll maybe be like 70% just like, tell me what this is or what does it do? And then 30% application. I know later in the semester, y'all will do like different experiments. Like you'll go through different experiments, and then he'll put that kind of question on the exam. Like, what does this give you? What are the results of this experiment? Stuff like that. But overall, very reasonable for lecture. Yeah. Any questions in the Zoom while we're at it? I'm also gonna send this copy paste one last time in case I didn't get any UINs. If y'all comfortable sending that. And I'm copying people's UINs down. Sorry. That's a cool UIN. Really easy to remember. Anyways. That's cool. Okay, cool. With that out of the way, y'all want to go over the practice problems? Let's do it. So I don't even know how to make a breakout room. I've never had to do this. Does anyone know? And you just want to take over so I don't waste y'all's time? Or you guys can just work on it as a class, because I don't really care about that small group stuff. Just work on it as you please. But we'll just do like all the in-person people will do it, and then all the online people will do it together. So how's that sound? Cool. So the way we'll structure it is just like you guys can either run through it one by one, or like if there's a really hard one, you can go over that one first and just like work your way through it, and then make sure everyone has like all the questions answered, because they are the problem sets themselves are very important. So how's that sound? Yeah, like that in the chat. It's really awkward over Zoom because it's like nobody wants to fucking work together in a breakout. It's so awkward, especially if y'all have your cameras off. Which is a hint, y'all should turn them on. But yeah, the Tuesday and Thursday sessions will be so much better. But yeah, go ahead, start working on it. Find a way to jump in. Somebody take a leap of faith, because I need to manage too, and I don't really want to micromanage y'all. All right, that's good. All right, pull out your shit. Come on, go. And I have my answer key, so if y'all have a question, just go ahead and ask. Because I want to be able to explain both. So it's actually eukarya and archaea for number 2, because eukarya is actually a branch off of archaea, as weird as it is, because you have your similarities, because the umbrella term is prokaryotes, right? So you have bacteria and archaea, with like, and they have a lot of similarities themselves too. It's like the circular genome for the most part with bacteria, and all of archaea has circular, I believe. But because eukarya is a branch of archaea, they're more similar, just like by nature of the phylogenetic tree. I don't know off the top of my head, I'll be honest. Like I took this class a while ago, so I don't remember something like that, but what is it? Oh my God. So which tree were you talking about? Oh, yeah. So yeah, this should be like this little node right here shows that these are two like similar. Cyanobacteria is bacteria, is the domain. Yeah. Yeah, another I guess another thing I should mention is that I will not know everything off the top of my head. And if you think I'm a dumbass because of that, so be it. It's whatever. I just like, it's been a while since I've taken this, but you know, I'm here to facilitate. I think I can get an answer if you give me like a minute or so. But yeah, that's all I want to say. All right, wait, let's see what we have in chat. Okay, okay. So number one from Whitney is correct. Okay, yeah. Number three is correct. Number four. Oh, is that true? I think that's just a more specific version of what I put. So for y'all, it's number four from Whitney. I just put that it was like all of like the distinctions you would make between prokaryote and eukaryote, so like nuclear membrane and organelles, which are different from compartmentalization, which I mentioned, which he mentioned can happen in prokaryote cells. Those don't count as like membranes, obviously. So yeah, number four, I just put like development of nuclear membrane and organelles, endosymbiosis and like oxygen and phototropes. So I think you guys are talking about like a cell eating a mitochondria, right? I think that falls under like formation of membrane-bound organelles. Yeah, that's just my opinion. I think you guys are on the right track, which is all that matters. How are y'all doing over here? Yeah, so prokaryotes can compartmentalize, I'm pretty sure. And it's not in the traditional sense, you know, like for example, eukaryotes like humans, we form like peroxisomes and stuff, right? Because you have like reactive oxygen species in our cells that you don't want just like flooding in and damaging like membranes and stuff. But for prokaryotes, I think how they compartmentalize is like lipid and protein rafts or like safe areas that are attached to the membrane. And that's where they'll carry out like different catalase reactions. Catalase reactions are like the ones that convert reactive oxygen species into something that's not as harmful. And all organisms need to do that, right? So yeah, they can compartmentalize, but it's very important to make the distinction that that is not an organelle, like a membrane-bound organelle. I hope that is correct. I'm pretty sure that's correct because he did mention that in chat or in class. It's number 6. Let's see what I put for that. Cytoplasm and cell wall bacteria. Yep, it's pretty good. So Patrick's number 6 is pretty good. Prokaryotes do have ribosomes, yeah. They're just free floating, similar to eukaryotes, yeah. But eukaryotes also have like ribosomes attached to the ER, but they also have free floating in the cytosol. How are y'all doing over here?
I should show the chat.
To y'all. Well, you guys do have a group meet, right? I would be careful with that though, because somebody might snitch. I shouldn't call it that. Somebody might sorry. Somebody somebody might tell the professor if they think that something is academically dishonest. And if it is, you guys could get in trouble for that. Well, yeah. So they're not worth a grade. So it's kind of like he's open to collaboration. And I emailed him and I'm like, can I go over these because I know you're not giving answers away. And he was like, yeah, it's like it's a good topic for discussion because they're so important. Doing well. So I don't see why he would be against y'all collaborating on that.
Can you go over number 8?
Yeah, give me a second, Haley. So if you go into office hours, he'll guide you to the answer. He won't give it to you. But he's really good about like making sure you get it. Yeah, cool. Could you repeat that, Haley? Which one?
Can you go over number 8?
Number 8. Yeah. So where does one molecularly or biochemically find the product of the question 6's DNA sequence? And I think it was actually question 7. Where does one DNA sequence? So I think what he meant by that is like where you would find the molecule from question 7, I believe it is, where it's talking about like the determined phylogenetic relationships. And if we look at the slides, what he was talking about in that case was the small subunit genetic information. So if we go down here, this is what it's talking about, right? And you have your protein side of your small subunit, and then you also have the rRNA. And where this is encoded is in like the nucleus, but when it's transported out, you would find, oh, so the product of the question 6's DNA sequence. So the product is the rRNA, right? And that is found in the cytoplasm or the cytosol, because that's where most of the ribosomes are. So what I put for question 8 was ribosomes are found in the cytoplasm. And I think that's what he means, but I don't think it's a greatly worded question. Does that answer? I also kind of like made it kind of convoluted, but does that answer your question?
Yeah.
Cool. Oh shoot, I should read the chat. Small you. What's up? My bad. What's up? Right. So obviously for the more important topics, he's going to spend more time covering them. Usually it's not as much of like the verbal cues. He'll like make it very clear if it's like something that you should know. For example, like if I go to the lecture notes, something like this is important. Okay, this is important. But I know there's like he'll like bold things basically. So like that's how you know it's important. I think it's safe to know everything though, because it's like it's a class where you should know the big topics more than the smaller ones, but he could take any piece of information from the lecture slides and put them on the exam. That's kind of a crappy answer for you though. I wish I had more. It's just to like have a sense for if he like covers something with more, like if he spends more time on a topic, that's what he thinks is important and it'll more likely show up on the exam, but everything is fair game if it's on the PowerPoint. Is that a good enough answer? Cool, cool. Did you want to say? Okay, cool. Cool. Yeah. Okay, so 7, 8, and 9 from Whitney.
Yeah.
Small RNA subunit. Oh shoot! Wait, could I get you to sign in real quick? I don't think you didn't get it. She did. Okay. Okay. Anyways, 7. I believe that's right. So number 8, we covered that one. I believe it's cytoplasm because that's where you find ribosomes, and then part of the ribosome is the small subunit. And ribosomes are not functional in the nucleus, I think is what you meant to say. So yeah, I don't think it'd be nucleus. The precursor would be found there, like the DNA that encodes it, but not the ribosome itself. No, the DNA would be in the nucleus. So, and then it encodes for the mRNA that I believe it's first an mRNA and then post-translational modification turns it into rRNA. The point is, you're never going to find functional ribosomal RNA in the nucleus, I don't think. And it's going to be really embarrassing if he says you can in the very specific instances, but like functionally, it won't be in the nucleus.
Yeah.
Oh, so yeah, it wouldn't be in the nucleus because there is no nucleus. Yeah, sorry, I'm thinking of just like more general ribosomal RNA. Well, it is, but that's like an organelle, or that's like a functional unit. Where it's found is in the cytoplasm. Yeah. Yeah. Of course. And then 2 subunits. Yep, small and the large and the small. Number 10, when he said large subunits, 5 and 23, Svedberg. And 16. Prokaryote is called 30S. So quick correction to Patrick's. I believe so 30S. Wait, am I whiling right now? So you're saying the small subunit, it's 30S proteins, and no, you're right. Yeah, you're right. Cool. Whitney's number 10, large subunit. Okay. Single-stranded looks double-stranded because it's compact. Yeah, so I put single-stranded, but it like folds within itself. I think I just knew that from prior knowledge, but if we take a look at the slides, we can probably see that wherever it may be. So yeah, so if you look here, you have these bases, right? And these are RNA because you see the uracil, but you also see right here, it starts with an A at the number 1 position, and then ends with an A at the 1500th 42nd position. So it's probably single-stranded.
Wait, so if it's starting
Well, there's no B.
okay.
This is the base.
Okay.
And what we're seeing right now is like, so okay, let's say you looked at this only, right? This little rectangular segment, you would think, oh, because they're pairing with each other, it's double-stranded. But if you look at the bigger picture, you can see that it starts at the 1500th base right here, and it loops around, and it makes this little folding like subunit. I forgot what it was called, but we did learn it. I should probably know this, but it's basically like folding in on itself. And then that's what adds function. Because if you take in biochem, you know, like structure dictates function, stuff like that. So like this and it's not a protein per se. So you can't say it's like tertiary structure, but the point is that it's folding in on itself,
So
but it is single stranded.
So with the 1390 area also folded on itself.
Yeah, so this is folded on itself, but it's not double stranded because it's just like the same segment, but it loops all the way back around and then connects with itself.
Double stranded about you like all of them would have a common. That's how we know that.
Yeah. It should. Unless it's like in the well, no, yeah, no, that's right. Should be right. And then the chat, I keep forgetting. It's so hard managing too for my first session too. I'm like a little newbie. Single stranded. So yeah, Whitney's correct on number 11. 12. The chat. Let's see if that's possible. I think that just makes it bigger overall. And I don't want to scroll up because people's UINs are there. Yeah, I don't know if I can do that. Sorry about that. So wait, sorry. Oh, all right, go ahead.
For enteric bacteria, do we need to know like specifically that it's in the gamma proteobacteria?
Probably. He made his own slide on talking about gamma bacteria. So it's probably important that you should know that these are like basically because he added proteobacteria and then like specifically gamma is probably important because this is like the most medically relevant. And it's just like it's just the most relevant in terms of like the proteobacteria. So know that these are, yeah, you should know all of these bullet points. Yeah, so yes, to answer your question, yes. Cool. Let me check the chat again. Number 12. What's number 12?
Oh, like I know that one.
So to narrow down on 12 a little bit, basically conserved regions in like different proteins, and it's usually like translational and like replicative units. Those are important because if you have like a tiny change in, let's say, like DNA polymerase 2, like that's going to completely fuck up your replication, right? And you know, if you can't replicate, you can't live. You can't grow, you can't live, stuff like that. So what's nice about the RNA is that it's highly conserved in a lot of regions. And the small changes are really good at dictating, you know, how much an organism has replicated or mutated, sorry. And then through those mutations, you have like divergence. So Winnie's correct in saying that the 16S ribosomal RNA is a good region to look at. But a better answer would just be the variable regions. What parts of this RNA? Yeah, so it's asking for the variable regions. And then the changes in those variable regions can help scientists create that phylogenetic tree. Yeah, it's a good answer, good answer. Question 13, oh my God, where's my mouse? It was a little hard for me because I'm not good at like doing these small things, but I put D and B for the first one, just because I saw like the fewest number of changes, and then I put C. Yeah, so Whitney put C as well. Yeah, I put D and B for the first part and then C. And then I just did that by like doing a really loose count of, you know, how many different nucleotides. But this is an example of like an application-based question. So yeah, I want to bring this up real quick. Question 13, where it has organisms A through D, that would be an example of like an application-based question, because you can't pull from your knowledge of like the lecture like what the answer is. You have to like apply a certain skill set. I don't need to explain to y'all what application is, but and I don't have it pulled up there either, but this is just like an example of that. So that would be like maybe like 30% of the exam. It's not as important, but yeah. What did y'all get for 13? Yeah, that's what I got. And then if you don't know how to do that, it's basically counting like the nucleotide differences. Obviously, if you have a more similar sequence, you're more similar of an organism. Yeah.
If that was on the test, would it ever, like, would the differences be more extreme to make it easier, or would it be like?
I really can't tell you. It's just what he decides to write for the exam. And a lot of times it's not. A lot of times you're gonna have those stupid test questions that are like kind of hard to differentiate. But it'll be similar to this because he's not gonna completely change up the format. Yeah. So if I remember correctly, it was like 25-ish multiple choice and then 4 to 5 for your response. And then the 4 to 5 for response usually are like a paragraph long.
Paragraph long?
Yeah. So make sure you have enough time. I would usually do the free response first because I'm like neurotic about that. Because you can guess on multiple choice and you can like speed run them. I should bring that up next. I don't remember, but I have my old exams and they have point values on them. I think free response ends up being like a 4 because they're weighted more because obviously like there's more thought that goes into them. So they're weighted more. I think they ended up being like 20 to 25% of the total exam, despite being like a 1/6 of the question. It's like I really don't want to. I want to play it safe, and I don't want to because he's writing my rec letter as well. So if he's like, oh, he's like cheating, he's helping like his students cheat. I really don't want to do that. I'm sorry, but I will try to make it like similar. And I'll even write a free response that's similar to one that's in the past. But I can't do that. I'm sorry. I'm really sorry. Proteobacteria, yeah. 14th correct from Patrick. 15. Gram staining, yeah. Quick quiz. What is like the substance that actually makes it purple? No, that's the fixing one.
Crystal violet.
It's crystal violet. Whoever said that, congratulations, you get nothing, but it is crystal violet. So the procedure is crystal violet to make it purple, iodine to fix it into the cell wall, and then I believe it's just generic like ethanol, alcohol, something like that, to wash away the lingering purple. Saffron, you'll learn in lab. I don't know what that does right now, I'll be honest. Well, my understanding is that the pink is just the washed purple. Like when you're adding a crap ton of, like, okay, let's say you have a gram positive and a gram negative side by side, and you just blast it with crystal violet. Both of them are going to look purple at first because there's a lot of purple on it. But when you wash it away after you fix it into the cell wall, it's not going to be as fixed in on the small cell wall because you have less, and you also have the outer membrane protecting it. So it's going to look pink because there's fewer connections of the purple. But the pink and the purple are caused both by crystal violet. I'm gonna check that because I really don't want to tell you the wrong answer, but that's my understanding of it. Can someone fact check me in the chat if that's wrong?
Yeah,
I'll go down.
yeah,
Oh, here?
yeah.
So yeah, all cells are purple at first, and all cells are purple. So once you wash it,
It has like white. And then they prepare something.
holy shit. Oh my God. Okay. What the hell is a counter stain?
He said that the red wasn't as dominant as the purple,
And like.
and that's why.
Really? Holy crap. Okay, I hope nobody left when I was making that distinction. Right. Got you. Okay. If they make that, or I mean, if they tell Dr. Garcia that I made that error, I'll try to get them their points. But shit, that's my first fuck up, and I just cursed twice, which is bad. But Oh, I feel really bad. Okay. Well, yes. So gram-negative is not just a low amount of crystal violet. It is this the saffron. Yeah. So know that. Good. We're learning together. It stains the cell wall of gram-positive. Yeah. I'm sorry. So it's like well, my understanding was that iodine will kind of lock it into place. And then the alcohol washes away all of, like the crystal violet that hasn't attached to anything. But whatever is on the slide always takes precedence over what I say, because I'm just a dumb undergrad.
So here's a question, I don't know what number it is, but it says, what if you forget to do the iodine step? Like what would happen?
Yeah, number 18.
Oh,
Shit.
Wait, so iodine.
I'm trying to manage like there is what's up? What's the question? What was the question?
it's 18?
18. Cool. Let me look at it. So in terms of technique used in question 14, if one had a culture of bacteria. Okay. Where's my mouse?
So it would turn, it would turn white as well, because there's no iodine. And then once they counterstain with saffron, I think it would turn. I'm glad that makes sense. 'Cause I was thinking,
So they're gram positive. So if you do the iodine, so my interpretation was that if you forget the iodine, it'll just like wash it away because it doesn't fix onto the cell wall. And if we look at this, the role of iodine is not said unless he does it down here. So it fixes to the thick cell wall, but not so much to thin. So if you remove this step, we can assume that if there's no iodine, there's no fixation to the thick cell wall. So what I wrote on my thing is that they would both look pink. Is that what y'all got?
yeah,
Cool, 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 cool. Then that is what the answer is.
Can you explain number 25?
25, got you. So if one found a spore, heated up, and found that it failed to grow into bacteria, what phylum would you guess the parent of the spore came from? So my interpretation of this question was like, basically, what kind of spores or what kind of non-reproductive like unit does do like the certain like phylum have, right? So your two choices are spores and endospores, right? And I think the primary distinction for me is that endospores are a lot tougher, right? So they have certain tolerances to dehydration and heat. So if you heat up a spore, versus an endospore, you'll find that spores will tend to die and then endospores will tend to survive it. That's why you have like certain autoclaving procedures if you guys are in like labs and stuff. It's to kill off like every single thing. So yeah, I put actinobacteria because that's where you find normal spores, as opposed to where you would find endospores, which I believe are found in Firmicutes. Let me check. Is that right? Are y'all okay? Y'all are not talking to me. Sorry. So yeah, so normal spores, or sorry, endospores are found in streptomyces, which are in the actino. Oh. So what makes? No. Sorry. Where is
It's
it?
a firmicute clostridium that forms the survival spores.
Right. And then yeah. So it's saying it's distinct. Sorry. So yeah, the actino are the ones that would have like the spores that would die whenever you heat them up. So for 25, I would put actinobacteria. But if the question asked if it survives, then you would put Firmicutes, right? Is that what y'all think? Maybe. Wait, what? What was the question? Oh, y'all got that. Right? Do y'all know what enteric means? Well, not it just means it lives within like another organism. So for example, like our gut microbiota, you have like the H. pylori and all that stuff. That's like an enteric species. Yeah. Because it shows up a lot, but I don't think he covered it in class. I don't know. Half the time I'm dozing off, I'll be honest. Cool. Oh shit, the session is almost okay. We're gonna cut this, but I want to take some time for y'all to tell me what you want. Because I'm assuming this is better than just like me telling you, like going over the lecture again and giving my own slides. Nobody really wants that. Do y'all like the problem set work, like study group stuff?
You could go over it, like number one, what you guys get.
Okay, yeah. Oh shit.
Yeah, we can all be on the same page as well.
Yeah, no, that would definitely add a little more structure. So I'll do that for next time.
Oh, yeah.
What else? What else? What else do you all want from these sessions?
So are you just gonna be repeating this session on Sunday? Or
Probably.
like, will it be new content? Okay.
Probably because I feel like the rain and all like the organization stuff messed up a lot of people's plans. But I want to make sure everyone gets like this information about how to succeed in the class. Stuff like that. So yeah, you don't need to come if you came today.
Is Candida like just a regular spore, and then there's like
Canidia, yeah. So
spores?
Canidias, I believe, are just
Okay.
regular ones.
Okay.
So Canidia means, I believe it's like a non-reproductive fungal unit. But the thing is, we're not in fungus yet. So I think it's just know that Canidia, I believe, means spore. In the case of like the first lecture, but you can always Google it, I think, if you want a better answer. So I'm not sure. But yeah. Did anyone else have like feedback? Because like, yeah.
Like completed the like questions prior to SI.
It would be nice, but I don't expect y'all to like do an assignment just for SI. It would help in terms of like discussion if y'all had it done beforehand. I feel like you can get a lot more if you have like some framework and you just keep adding onto your answer. And it always sucks being like left behind when everyone's talking about stuff. But yeah, so yeah, it's nice if you have your worksheets somewhat done. Cool. Thank you so much.
So he just released a new worksheet, right? So when are you gonna go over that?
I'll have it done by the Tuesday session. I think is fair. Yeah. And I'll send it. I'm planning on sending out reminders or announcements every Monday at like 10. So you'll know exactly what we're covering on each day. Thank you. Yeah. Have a good one. Be safe.
Yeah.
But yeah, if y'all don't have anything to add, you guys can dip. But again, like this is just for me to have an idea of what y'all want. I'm just here to listen for now. So if you have something that you want me to do in SI, please just drop it in so I can make these sessions actually worthwhile.
Thank you.
Of course. Thank y'all for being patient. Have a good one. Everyone dips so fast from the Zoom.